Yeah, so in today's lecture of botany, we are going to see the marker enzymes and their role in identifying the special subcellular organelles. So basically, these marker enzymes are used to identify something, as the name suggests, the marker. So these are a very brief brief introduction to the several kind of markers here. Like markers can be the type of biochemical, morphological, and molecular. So as I was telling you that what is marker and what are the function of marker. So marker is present inside the cell organelles as we use them just to identify some special kind of subcellular organelle, or sometimes we use them to find the activity inside the cell. So this marker, if we are talking about the very common type of marker, these are the morphological markers. So actually, morphological markers are based on by identification majorly. So if we wanted to, if we are seeing something, or on the basis of the viewing, or just by scanning something, whatever we thinks, whatever the analysis we got, that is based on the morphological markers. Like we introduced two kind of effect of gene inside a cell. Like one effect of that gene is they can give the yellow color. Second gene effect that they can give the red color. When we got a flower, after emerging those both of the gene inside them, the color of flower what we got in this experiment, we can only by seeing them we can tell what kind of gene showed their effect inside them. So on these bases, on these pattern, or sometimes the height character you can analyze. So by just seeing some shape, size, color, surface, these are the things which can be analyzed just by their outer structure or outer structural studies. They come under the morphological analysis or morphological markers. Second markers here come the molecular markers based on DNA, and they will be going to tell us the information on the basis of two methods. What are these two methods here? Based on the hybridization, that can be the RFLP, mini satellite, and microsatellite based hybridization. Second one is based on PCR. So on the basis of PCR, we can do the molecular diagnostic like AT-PCR, SCOT, RAPD, EST, ISSR, STS, AFLP, SCAR. So these are the techniques which are based on the PCR. And why we are using PCR here? Because PCR is determined for giving us the amplified results. So anything which is very tiny and we cannot analyze its in individual state, at that time what we need, we are going to need PCR with. And the major thing what PCR can do, it's amplify. They can amplify, amplification work can be done by the PCR, and after amplify we got something in bigger in picture, so we can analyze it. So these were the things, these were the markers that are dependent on the molecular analysis. Second, here the biochemical, they are dependent on the isozyme protein binding pattern. So biochemical isozymes means they are depend on some of the enzymes, and they are going to tell us about the cellular activities. Like we have a lot of cellular activities present inside the cell. Every organ or every organelle inside the cell, they have a unique function to perform. They have a unique location. They have a unique function. Like the organelle talk about mitochondria, peroxisomes, cytosomes, lysosomes, cytosol, or the other thing Golgi, ER. So they all have the different functions to do, and also their enzymatic activity are also specific to them. They cannot be, enzyme activity of each and every organelle. They cannot be co-op with one another. They have to be very specific to each one of the organ. So let's begin with what are enzyme markers and why they are needed. So what are enzyme markers here? Enzymes are highly specialized complex protein that aid chemical changes in every part of the body. For example, they help break down food so your food can use it effectively. So basically, if we talk about the enzymes, so they are a protein supplement, or you can say they are the different structure of the protein, or if we see in the basic manner, they are the amino acids having some special structures and function specified to them. So they are responsible for the chemical changes in every part of the body. Not they are not responsible for physical change. Whatever the change they have been made, most of them are irreversible. So, for example, they help break down the food. Like there are several cycles happens inside our body: Krebs cycle, pentose phosphate cycle, glycolysis, gluconeogenesis. A lot of cycles happening inside our body, and they are affected by these enzymes. They are catalyzed by these enzymes. They increase their rate of reactions. So this is a major function. You can say these are the things which really need for doing all the cycles, all the metabolic activities inside our body. So they also help your blood clot when we got any cut on our hand or leg anywhere on the body. So they also help us to make the blood clotting and stop the bleeding, and they are present in every organ and cell in your body. So in our body, in every organ, in every cell, even in the cell organelle, they are present everywhere. Enzymes are necessary for body to function properly. So if they are not present inside our body is not going to function appropriately. They are the very needy situation in the our body to do their metabolic pathways. So, enzyme markers or blood tests that analyze specific enzyme activity in the body. So, if we do simple blood tests, they are the diagnostic test, and on the basis of this enzyme activity inside our blood, we can analyze some kind of disease. So, some inherited disease or condition can cause these enzymes to stop working or to be less efficient. So there can be anything like inherited disease means in which DNA are involved. Maybe DNA has to be like this in their previous parents and in their parents only. So their parents maybe have the code for like some of the enzyme has to be produced in a less manner by their coding pattern. So maybe this kind of gene that has been forwarded into their offsprings and showing a dominant effect. So on the basis of their study, on the basis of the enzymes study, we will able to know what is the basic problem inside the body, inside the cell, inside the organ. Monitoring the rise or fall of enzyme levels can aid in the diagnosis of variety of conditions. So, when we study these conditions, we are going to able to find several things about our body conditions. Isolation of any organelle require a reliable test for the presence of organelle. So, when we do the isolation of any organ. Like we wanted to isolate the mitochondria, we wanted to isolate chloroplast, lysosome, cytosol, anything which we wanted to isolate. So typically it is done by following the activity of an enzyme that is known to be localized exclusively in the target organ. So how we can do it? Typically what the first thing we have to know, what the thing we have to do, that we really have a we really should have a proper knowledge about the enzyme activity that is present in that particular target organ, because on the basis of their enzyme activity we can be able to locate them, we can be able to see that how they are functioning inside the body. Such enzymes, which are responsible for identifying them, are known as marker enzymes. Okay, so this is our topic, and that's why we are studying marker enzyme because they are so important. For example, the enzyme acid phosphatase that cleaves terminal phosphatase group from substrate and has a pH optimum in the acidic range is localized in lysosome. So now we got enzyme and they are doing their activity by cleaving the dominant phosphate group from its substrate in a very high acidic range. So by seeing these kind of activity, we can localize the lysosome. As we just seeing, oh, this kind of function is happening there, so we clearly know that this is a lysosome. No other can do this kind of function. So that's why we are calling them the marker gene, because if they are showing their activity in a particular place, we will be going to identify what place it is. While the enzyme succinate dehydrogenase is localized in mitochondria, and if we got another enzyme that is succinate dehydrogenase, and they are showing their properties, so we'll be getting know that this is the enzyme of mitochondria, and if the function is doing there, that possibly the site of mitochondria. By monitoring where each enzyme activity is found during a cell fraction protocol, one can monitor the fraction of lysosome and mitochondria. So basically, when the fractioning happens, when the goes under the lysis, then these all situation can be monitored and can be studied. Microenzymes also provide information on the biochemical purity of the fractionated organelle. So after the fraction, when we do the cell fraction, in the process of cell fraction, cell walls will slice and burst it out. So busting out, or breaking down of the cell wall, they will going to release several all the cell organelle present inside them. So these marker enzymes, they also tell us, or they are also able to tell us their biochemical purity. So biochemical purity means, like, if they are having some unwanted thing inside them, or if they are not doing their process in an efficient way as there has to be. So this thing can also be, this will can also be come under consideration by using the marker enzyme. So the presence of unwanted markers enzyme activity in the preparation indicates the level of contamination by other organisms. So you can also see the contaminant portion. If anything has, you look like that this is a portion that can cause disease. This is contaminated. This is enriched. So anything, the bad and good, you can know about that particular portion by using the marker enzyme. While the degree of enrichment for the desired organelle is determined by the specific activity of the target marker enzyme. So normal enrichment of that desired organelle, that can be determined by the specific activity, because these marker enzymes, they are specific to every organelle present inside the cell. They cannot, like, you are using a this marker enzyme that worked on the mitochondria and now it is going to work on the cytosol or some other organelle. They did not work like that. If they are working that, like, they will be create some confusion. How you will going to differentiate that this activity is inside mitochondria or cytosol? So that's why these enzymes, they needed to be very specific about their working process. Although marker enzymes reveals much concerning the purity of the organelle preparation, electron microscopy is generally used as a final step to assess the preparation purity and morphology of isolated organelle. So after when we treated them with the results, the final results where we can see it under the electron microscopy, or with the help of electron microscopy we will be analyzing much of their state, how their morphological analysis. If we just wanted to see the surface view, we can use a SEM, scanning electron microscope. If we wanted to see their internal structure, how they're working from internal side, we can go for TEM, transmission electron microscope. So this is our goal. These are some organelle, okay, and they are the enzymes or substrate which are going to use for their enzymes, like nuclei, DNA, RNA. For the mitochondria, basically succinate dehydrogenase, but sometimes cytochrome C oxidase too. For the lysosome, the enzyme we are going to use, that is beta hexoneamidase or beta galactose acid phosphatase. Maybe that also can be used. For the endosomes, first reddish peroxidase that can be used for peroxysomes, the catalase that can be used for identifying Golgi. Actually, Golgi have two states, like the cis and trans state. So the cis and trans state, so Golgi apparatus in the cis level, we can use by the marinase for the sorry for the trans level it can be used by the manoxidase, and for the cis level that can be obtained by the galactosyl transcripts. Endoplasmic reticulum, if we wanted to analyze the activity and their location by the enzymes, glucose 6 phosphate is a basic enzyme. Cytosol by the lactate dehydrogenase that can be identified. Most of the function happens at the cytosol, but lactate dehydrogenase are the one which plays as an intermediate position. And due to having intermediate position inside them, they possess many enzyme activities inside the cytosol. So just by seeing the working of lactate dehydrogenase, we will be able to see what kind of activity is going on inside the cytosol. Now, if we take a condition here, like your doctor test for enzyme markers or a routine blood test to help uncover abnormalities. In some cases, you may need to take a test multiply times over the course of several days to measure changes over time. So, if sometime when we go to doctor and tell him about your problem, tell her or him about your problem, at that time, what happened? They always refer us to do some diagnostic test, like the blood test, thyroid test, lymph test, any test which they think has to be associated with our problem. So when we are going to test it, so you know what the major tests they are going to do? They are doing the tests that are based on the marker enzyme, that are based on the thing, or you can say marker enzyme, in which marker enzyme going to tell them what kind of problem is there, is everything inside the cell is doing properly or not, or any kind of problem is there. So these things can be tell us by these tests to our body. So, what are the common type of enzyme markers inside the? You can say it's a health problem when we do some test. This is based on this. This is based on the testing of disease. So, in this CPK iso enzymes, they are the type of the enzymes marker which we are going to use in so many detection techniques. So the CPK enzyme test measure the creatine phosphate. Okay, so this is creatine phosphate kinase. So this CPK is called creatine phosphokinase in the blood. So we just prefer it to call CPK only. CPK enzyme are in the heart, brain, skeletal muscle, and normal CPK levels are vary by age, gender, or race. So now you can see that on the basis of CPK inside us, okay, and CPK are responsible us to telling the more about heart, brain, skeletal muscle, and these are the important functional zones of our body. That's why we are taking the example of such kind of important common enzyme marker. And this concentration, or you can say the amount of this CPK, that also vary with the age, gender, or race. Female or male, they have different at the age. Child have different. Adults have different. Or the one like 50s to 16 and old ones have different races, they can be vary. So each laboratory may have minor differences in reference age as well. In journal, a CPK level approximately 200 units per liter. So some can be tell us like the 180, or some can be tell the 220, but not more than that variant can become. So approx gene, or you can say approx enzyme quantity that has to be in our blood, that should be the 200 units per liter or less. Considered normal in an adult. So normal, it will be okay. This is total CPK level in your body. More specific testing can be done, but this is not. routine. So that can be done several times, but not on a routine test. So CPK1 residues mostly in the brain and lungs. Increased level of CPK1 can be due to, like, if we are having the more enzymes of CPK inside our body. So what are they caused of? Why were the reasons when we do a CPK test and we got increased CPK? So what are the problems which can be present inside our body? Are first the brain cancer, second the brain injury or stroke or bleeding in the brain, pulmonary infractions, which is the death of the lung tissue, seizure, electroconvulsive theory. So convulsive theory. So these are damaging. You can see these are the major problems inside the body. So CPK levels should be maintained, and by just knowing the enzyme CPK, we will be going to identify way more diseases inside our body. So if it is normal, everything is normal. But if it is increased, then these are the problem. But now we'll see CPK2 level rise following a heart attack. Okay, and CPK2 level they can be following a heart attack too. Increased level of CPK2 may also be due to open heart surgery. Why? What are the reasons of having CPK2 level increase? Open heart surgery, inflammation of heart muscle, heart injury, defibrillation, electrical injuries, chest compression after cardiac arrest. So these are the medical terms which are having a serious kind of case condition, like the heart injury is not simple, electrical injury. They are can cause a very high level of CPK inside our body. So the cardiac arrest. So these are the things. If they are not normal, so the CPK level can be high inside our body. Now, the third, high CPK level can be a sign of muscle stress, uh, crush injury, or injury due to muscle damage. That's just called dystrophy or inflammation, intramuscular injections, electromyography, which is a muscle and nerve function test. So these are the thing which can be cause high CPK3 level. Like this test even can cause that to recent surgery, scissors, strenuous exercise. So these were the cause who can increase the CPK3 inside our body. Now we're going to see the heart enzymes, how the heart enzymes work to localizing. Some heart enzymes slowly enter your blood if you have a heart attack and your heart is damaged as a result. So basically, these heart enzymes, if they are going to enter, enzymes slowly enter into your blood when when you had a heart attack. A general test of emergency room patient with heart attack symptom is a test for the presence of certain proteins in your blood. So the heart attack cause in when we see that these certain kind of protein increased inside the blood. So, what are the checks which doctor is going to do here? A doctor may check CPK2, also known as CKMB, and this marker is highly specific for heart muscle injury and rise rapidly during a heart attack. So, if they are going to see a heart attack, whether it's CPK2 increase or any other problem. Normal CKMB should be between 5 to 25 international units per liter. Okay, that should be very less, but if it is increased, they can cause a heart attack situation. Okay. The preferred marker of heart injury through is a protein called troponin. Okay, so troponin, that preferred marker which caused by the heart injuries, and troponin should generally be less than point 0.2 nanograms per milliliter. Okay, this level takes longer to rise in CKMB, but the protein stays in the bloodstream longer. So this level, they will take longer to rise than CKTV, means they do not rise like the CKTV MB rise, but these protein stays in the bloodstream for many times, and they can be give problem at any time. So the liver enzyme, so this was these two, okay, CKMB and troponin. They were the hard enzymes. Now liver enzymes. Elevated liver enzymes may be due to inflammation of or damaged liver cells. Usually, elevated liver enzymes are related to an acute injury or a process that occurred over a short period of time due to now, they are this liver enzymes. So how they elevate it? So they elevate it due to inflammation or damage of liver cell. If any liver cell it is going to damage or inflammate it, then only these liver enzymes going to pop up. And what are the reasons of that? Prescription medication such as statins. So some of the medication even can do that, or over-the-counter OTC medication such as acetaminophen, or you can say Tylenol. So the Tylenol are also they cause something to cure, but also they have some major side effect for mostly on the liver. And many medications, many drugs have common tendency to show the bad influence on the liver, and this will cause these kind of problem here. Alcohol consumption can also damage the liver cells. Heart failure or heart attack can also damage the liver cell. Liver disease such like hepatitis, fatty liver disease, cancer, cirrhosis, obesity. These are the problems, these are the common problems which can cause the liver enzyme to increase from their level. Celiac disease, which is a digestive condition, they can also cause the enzyme activity to be induced inside the liver. Some viruses, such as cytomegalovirus infection, hepatitis A, B, C, B viruses, mononucleosis, and Asper B virus, EBV virus. So these are the viruses also. If the multiple growth of the virus can happen inside the liver for a long time, so they can also create this kind of enzyme activity concentration increase. Muscular diseases such as muscular dystrophy or polymytosis, ICM, or lack of oxygen going to liver, such as during cardiac arrest. So these are, a lot of measures, a lot of things which can cause the basic activity of enzyme, or you can say the level of inside of enzyme inside the liver, and this will going to cause several problems inside the body. So how are enzymes marker test performed? So how we can perform the this enzyme marker test? Test is a routine blood test, okay? It's normal, we don't have to do something else or big. We just go for a blood test and take place in a laboratory. No fasting or special preparation is necessary, nothing new has to be done. Tell your doctor before the test about all prescription and OTC medications and supplement you take. So anything additional from your normal food, if you take like some prescribed medicine, OTCs, medicine, or supplements or drugs, anything what you think is not normal and you are taking it. So this is the primary thing you have to tell your doctor. So that then they will do the test according to those parameter, like what they have to ignore and what they have to follow from that. So a blood test involve the following step. Now the blood test which is going through this process, they are going to have some following step like healthcare provided with uses antiseptic to clean a small area of your arm. So first just clean your arm from where the blood has to be taken. Usually the inside of your elbow or back of your hand. So they can be taken behind inside of your elbow or back of the hand. They will then wrap an elastic band around your upper arm to create pressure and make it easier to access the vein. If sometime vein is not accessible, we can do this. After that, they will insert a needle into your vein and blood will flow into a small vial. You will likely feel the stick of a needle or a stinging sensation. So this is just as simple as to take a blood sample. After filling the vial, the healthcare provider will remove the elastic band, obviously. They will place a bandage over a puncture site and send a blood sample to the lab for analysis. The procedure should take only a few minutes to tell about all the enzyme activities or all the problems associated with your blood. So, what are the risks they are associated with enzyme marker tests? So, your arm may be sore at the puncturing site and you might have some mild bruising or teeth throwing. That's okay. Most people have no serious or lasting side effect of blood test. Rare complications include bleeding, lightheadedness, fainting, infection. Honestly, nothing happened. So these were the things when we analyze the marker. So what you have to remember in this whole lecture is this point. That markers are the enzymes which are responsible for identifying a particular organelle inside the cell. And these organelles are specific for the enzymes and their substances. Like nucleic acid, they will be deciphered DNA, RNA, or I just told you all the things, this table that you have to be remember, and you have to remember that why we use a marker enzyme to identify something, if you wanted to see what thing present at what place inside the cell, and secondly if you wanted to see their activity. And for coming to this level, we have to go through with like 2 to 3 steps. First step is for the lysis of the cell. When we break down the cell, after the lysis of the cell, what we got, all the cell organelles. And after getting all the cell organelles, we have to go through the process of differential centrifugation. What happens inside the differential centrifugation? You needed to be go one by step one. I will show you if I had a PPT of the centrifugation right now. It will be easy for you to remember again. Give me 1 minute. So this one. So for obtaining different kind of for obtaining different kind of cell organelles, you have to go through this differentiational centrifugation process. So when we are going through the differentiational centrifugation process, on each and every step, here we got particular cell. This is a cell homogenate that we make, and after giving some low speed centrifugation, we got a palette, and in this palette we are going to the nuclei and cytoskeleton. And on the basis of these specific marker enzymes, we are going to see are they working properly or not. After this, that we giving some medium speed centrifugation, and we going to get the palette 2, in which we are going to get the mitochondrial lysosomes and peroxisome. Third stage, we got the supertent here, and again both them let them enter the centrifugation. And this palate third, they will be going to have microsomes and other small vesicles. So now the enzymes which are related to these microsome and vesicles, we are going to analyze them. And after the last one, we are going to very small and very light particle, ribosomes, viruses, and large molecules. So basically, we need a different degree of centrifugation to obtain these subcellular organelles. And for watching with how these subcellular organelles are working, we go to do those micro or marker enzymes. So that's what we going to need before we test for the micro marker enzyme testing. First, the lysis of cells. Second is this differentiation and centrifugation, and third we go through for marker enzyme to see their activities. Okay, so this is all about how the marker enzyme work on the subcellular organs. I hope you understand it. If still you have any problem,